So yeah, I'm really excited to present my work today. I would like to start by thanking the organizing committee. So I think what you guys are doing with the synthetic biology community in Peru is really amazing, and I hope it can carry on for the upcoming years. It's great to bring people from the community together, not only from Latin America, but from everywhere else in the world. I think what we're doing with iGEM and all these kind of competitions that you guys mentioned in the Malta and previous speakers are something to keep doing to build the synthetic biology community. So now I'm going to get into the presentation of the work I've been doing here in Montpellier in France for the past couple of years in the lab of Jérôme Bonnet. And this work is called Cell-Free Two-Hybrid platform for rapid protein binder screening and more. All right. So before I get into the kind of basics of the project, I would like first to show you guys what are our interests in the team and what we are doing in the lab, more specifically. So we have multiple areas of interest. Some people work more on biosensors. So you would hear from the next speaker, Anna Zuniga, who is going to talk about what she's been doing on biosensing purposes. And what you can see on this schematic is bacterial cells. So we mostly work with E. coli, but we also work with other microorganisms like lactobacilli. And we see, as is often used in the field of synthetic biology, the analogy with computing or electronics. So we see bacterium as small devices that we can try to reprogram to our advantage. So we can reprogram them to sense specific cues. They can be chemical cues, like small molecules of interest. So maybe I'll make a laser pointer so it's easier to see. So you can sense molecules of interest for diagnostics, for example, like bile salts. So you can re-engineer the DNA of the bacterium, introduce some new parts, and tell the bacterium to produce a specific output in presence of these chemical cues. The other part of the lab is focusing on bacterial therapies, but these two parts are actually quite linked together. So you can imagine that if we engineer a bacterial cells that can sense specific inputs for example, lactates, which is present in tumor microenvironments, and link that to the production of therapeutic molecules like nanobodies, interleukins, cytokines, or de novo binders, we can try to use bacteria as smart delivery vehicles that would be able to target precisely therapies to the tumor site. And so it's in this context that we started with the design of de novo binders. And this is why we came up with this new method to screen these binders. Okay. Why don't you want to work? Okay. So it started with the AlphaFold revolution. So just to put a bit of context before getting into the details of the project, it started with the AlphaFold revolution, and you guys all witnessed, and we all witnessed, the fact that now from the amino acid sequence alone, you can get a predictive model of a protein structure in 3 dimensions. And so this was not possible before, or not very accurately. And this kind of changes everything in the way we are approaching problems. And other tools complemented this revolution with inverse folding, for example, which enables the opposite direction. So starting from a given structure and predicting the most likely amino acid sequence that will be able to fill in this structure in the lab. So this kind of changes everything for protein binder design, more particularly, but for many other topics as well. So now we can with a certain confidence, design proteins entirely de novo, which are proteins that do not exist in nature, and give them specific tasks. And let's say I want to bind this particular target protein on this particular epitope, and you give that to these new programs like RF diffusion, Bindcraft, or other kind of methods that are similar. And basically, these methods are going to compute parameters and do modeling and come up with possible design that would fill in these functions. Jesus, this is annoying. So now we have a shift in the paradigm of protein binder discovery. So basically, before the AlphaFold revolution, we had what we can call a wet lab first era, in which basically everything, all the magic was happening in the lab, where we would screen massive libraries of candidates, of candidate binders. It could be single domain antibodies, for example. And then we'll hope to find a good antibody that would bind a target sequence. So this obviously would take a lot of time, a lot of resources. And at the end of the process, we could do a bit of rational design once we had the structures to try to kind of infer better affinity or better specificity and so on. Now, what's happening is that everything is kind of shifting to an AI-first era, in which in silico designs are being done on the computer, and most of the filtering and the screening actually happens in silico. So you can design thousands to millions of proteins, and then you have metrics that you use to rank these designs. So let's say you don't need to screen all these designs. You just need to take the best ones, let's say 96, to fill up the whole 96-well plate. And then you can start with that and maybe do a bit more iterative rounds and try to learn from the design that worked and the design that didn't work to build up new models and come up with better approaches. The methods, though, in the lab to screen these binders have not really changed since then. We still rely on all the methods I'm showing here on that slide. There is, on one side of the spectrum, you have biophysical methods that can be SPR, so surface plasmon resonance, biolayer interferometry, and so on. So these methods are very resolutive. You can get access to association, dissociation constants, and so on. They're quite expensive and very low throughput, though. So for screening 96 binders, it's a bit too much. On the other side of the spectrum, you have display methods, which have been developed to screen libraries of billions of candidates. So they're very powerful, but they seem a bit overkill to only screen 96 binders. And obviously, experimentally, they still require a lot of steps. So if you do the experiment for 10,000 binders or for 96, you'll still require the same amount of steps. So when we looked at this, once we were interested in designing binders, we wondered whether we could design potentially protein binders that for certain purposes, and we wondered whether we could develop a method that would be fast, affordable, experimentally simple, but compatible with medium throughput screening, which is not possible with the methods at the minute. So we turned to cell-free transcription translation systems for protein binder screening. And I'm going to explain to you what cell-free is and why we turned to cell-free systems for that purpose. So cell-free systems are essentially bacteria cells that we grow. So we grow E. coli cells to a certain optical density. And once they reach that density, we harvest them, we wash them in some buffers, and then we lyse them. So we break the cells and we collect all the soluble fraction. So this we call lysate or extract. And this lysate, we mix it with an energy solution that is composed of many cofactors, amino acids, and so on. And with your plasmid DNA containing your gene of interest that you want to express. And once you mix all these three components together and place the cell-free reaction at 30 or 37 degrees, it's going to start expressing your protein. So it's going to start transcribing and translating in vitro. And this is great because you can do that very rapidly. And it's an open system. So you can add multiple DNA sequences. There is no antibiotic resistance, origin of replication compatibility here. You just add everything you want into the cell-free system. Now the trick for binder screening that we used is using an E. coli strain, so E. coli BL21 Rosetta, that has been deleted for REC-BCD. So REC-BCD is a gene operon, so a set of enzymes that can recognize and degrade linear DNA in E. coli. And this is originally a defense mechanism against phage invasion. So phage infection typically injects linear DNA into the bacterial cell. And so REC-BCD can really rapidly find these linear DNA ends and degrade the DNA. But because we deleted these genes, now it means that we can use linear DNA directly into our cell-free system without it being degraded, And this is really important because it makes the system much faster. So in summary, cell-free is quite inexpensive. It's basically E. coli cells that you grow and lyse. We work in very small volumes and 300 NT4 well formats. We use 5 microliter reactions. It's very simple. It's just mixing reagents together. So the protocol is very simple. It's similar to what the PCR setup is. And it's linear DNA compatible. So it means we can do things by ordering gene fragments and directly add them into our cell-free system without any cloning, culturing, or sequencing step. Now, to engineer a cell-free two-hybrid system, we took inspiration from nature, and we looked at, in particular, this lambda phage protein called C1. And C1 in nature is responsible for the transition and the tight regulation of the transition of the lambda phage cell cycle between lysogenic and lytic phase. So C1, as you can see on that structure, works as a dimer. So it forms homodimers. One of the monomers here is grayed out in the structure, and the other one is colored. And you can see that at the N-terminus, you have a DNA binding domain in gold, which is linked to a C-terminal domain by a flexible linker. So this linker in magenta is highly flexible and very long. And this C-terminal domain is responsible for the dimerization of the protein. So when you have C1 expressed in cells, essentially is going to form dimers, bind to specific DNA operators. Here I represented them in gray boxes, so operator 1 and 2, and activate this specific promoter by priming the RNA polymerase. So we designed a specific reporter plasmid that contains this particular promoter with the operator sequences, and downstream of this promoter, we put a GFP gene. Now we have a linear DNA cassette that encodes for C1, and our first experiment was to mix these guys into a cell-free reaction. And if everything goes to plan, we should have C1 expression forming homodimers, which will activate transcription and production of GFP. If you look at the reaction over time, this is what it looks like. So on the y-axis, you have essentially GFP intensity, so fluorescence intensity. And on the x-axis, you have time in hours. So you see that very rapidly, we start producing protein only in the presence of the C1 gene. If you don't put C1, but only the reporter plasmid, you don't have any GFP expression. Now, obviously, we're not very much interested in the wild-type version of C1. we want to study protein-protein interactions and develop a method for binder screening. So we need to somehow re-engineer that system to our purpose. So to do so, we modified a bit the C1 protein, replaced the linker by glycine serines, and then the C-terminal domain by a target protein and a binder protein. So now the dimerization of C1 and its binding to the DNA is conditional to the binding between the target and the binder. So to test this in a very simple format, we chose two couples of proteins that are known to interact together. These are coil-coil proteins from the Gerala lab in Slovenia. They are very simple domains. They are alpha helices that interact with each other very specifically. So P1 specifically interacts with P2, and P3 interacts with P4. So we kind of challenge these guys all against each other. And so that in our assay at 8 hours, we had really good signal only when the matching pairs were together. So it means that our cell-free system, so C1 PRM system, is functional in cell-free TXTL, and that the engineering of C1 enables the detection of specific protein-protein interactions. Now, we went a step further and kind of tried different protein-protein interactions. So we tried nanobodies, de novo binders, monobodies, or the spike tag spike catcher system, which forms a covalent bond between the tag and the catcher. And in all these tests, we could always detect interaction only when the target and the binder were present in the reaction together. To understand more the limits maybe of the system or the potential of the system, we also did an experiment in which we fixed the target. So this is the mCherry that you can see in pink, and 3 different binders binding to mCherry, but they bind to mCherry to different interfaces. So you have a dopin and 2 nanobodies. And the point of doing this was to know whether, so you have to remember that there is C1 DNA binding domain fused to these guys in our assay. So we wanted to know if the orientation of the N-terminus mattered, or if with the flexible linker it was okay for the binder and the target to find themselves in solution. And what we found is what for the 3 different binders, we could find interaction, and obviously for the negative control that doesn't bind mCherry, we had no signal. So overall, this confirms that our cell-free Two-Hybrid methods can detect a broad range of protein-protein interactions and actually tolerates diverse spatial conformations of the proteins. Now, if we kind of make an ideal screening workflow, it will look like this. So the first part will be, I guess, variable in terms of timings, and it would be the binder design. So you would choose your favorite protocol to design binders against your favorite target protein. Then you'll order DNA fragments, so linear DNA fragments that are fused to the DNA binding domain of C1. This usually takes 2-3 days to arrive to a lab. And then you do the cell-free experiment. So in these conditions, usually we do experiments with the DNA of the binder alone. We also do the target alone, or the binder DNA and the target DNA. And so the binder alone actually gives us some hints on the possibility of the binder to form homodimers or aggregate and so on. So it's also useful to have this feature. So we wanted to test this workflow and to kind of benchmark it against other methods that exist. We took kind of existing sets of binders. So we took binders that were validated in the RF diffusion paper by Watson and colleagues from the Baker Lab. And in this screening campaign, they validated a set of binders against MDM2, which is a human protein that regulates p53 inside cells and is often kind of causing cancer development and some forms of aggressive cancers. So it's a very well-studied target. And in that case, the designs that were made using RF diffusion were very successful. So here on this graph, I'll try to simplify it in the first place, so you guys can follow along. So you see with the diamonds, the black diamonds represent the scores that Watson and colleagues got using their screening assay. So their screening assay is based on single point biointerferometry measurements. So they basically purify all these binders. They put them in contact with the target using the BLI equipment, and then they measure the diffraction of the light that is produced by the binding of the binder on the target. And so that gives them a score, and here is the normalized score from 1 to 0. And you can see here the highest score is potentially the highest binding protein. And so they decided that all the designs that were below 0.5 of normalized score were not really good binders, but they classified them actually as non-binders. So this is the squares you see at the bottom here. In gold, it's binding designs, and in kind of gray, it's non-binding. And obviously the other ones that we tried are true negative control from a different binding set. So these binders have actually been designed to target a different proteins that is completely unrelated to MDM2. So these are true negative control in the assay. So then we did the workflow I just presented you, and within one day, this is the results we got. So you can see here in the blue bars are actually the binder with the target. The gray bars are just the binders alone. And you can see that in presence of the target, we have many hits for all the binders. So in total, we could find 21 out of 24 binders that were found by Watson AR. And we had also 0 false positives in our screen, meaning that it's actually quite good for a very simple screening method. So we obtained all this data in hours rather than weeks of work, and using very small amounts of cell-free reactions. So we're quite happy with these results. So to sum up on this part, so cell-free Two-Hybrid enables a cloning-free validation of dozens of de novo design proteins within less than a day, basically. But we were curious, and we wondered whether we could have more than a binary answer, like binder, no binder. Can we have actually hints on the affinity of a given binder for its target? Can we rank the binders from the most promising one, so with the highest affinity, to the least promising ones? And this would be very handy in a screening campaign, because you could imagine in one single screening step, you could already isolate the most promising candidates for then further characterization. So to do that in kind of a controlled fashion, we did a test by doing alanine scanning mutagenesis on a specific binder, which is called HA4. HA4 is a monobody, and it binds to the SH2 domain of ABL1. So you see in this cartoon, we have the interface between HA4 in blue and SH2 in gray. And a few residues are involved in this interaction. And so we replaced these residues by alanine residues and kind of measured the impact of that on the signal we get in the cell-free assay. So you can see that mutating the first tyrosine 35 does not impact much does not have a great impact on the overall signal that we obtain. However, mutating the tyrosine 36, which is much more involved in the interaction, completely abolishes the interaction between HA4 and SH2. And then the next residues over have kind of an in-between effect on the binding. So if we try to compare these findings in kind of comparing the activation levels that we get in our system compared to delta G values published in a previous paper, we see that we have a quite good correlation with what they got using much higher end equipment and techniques. So to check whether this was just luck or if we could kind of apply this to as a rule for kind of selecting best binders from the worst ones, we tried a different couple. So here is BCL2 and LD3, which is a de novo binder. So BCL2 is a human protein, and LD3 is de novo binder that binds specifically to BCL2 through this kind of residues I show you here. So we tried to mutate these residues by replacing them with alanines once again, and you can see that this time we measure signal activation in our system compared to KD, so affinity. So the lowest KD means the better affinity. And you can see that here the correlation is not that great, but we can still rank from the top binder to the weakest one with our system, which is already quite good. So we can have, let's say, an insight on the impact of individual mutations on the binding affinity, which is not only interesting for selecting the best binders from the screening campaign, but can also be used in kind of a second step to validate the interface of that is predicted when you design your binder in silico. You have a predicted model, but you're never sure if the interface is respected. So if you mutate residues around the interface and replace them with alanine, you can ensure that the model is actually true. So now we wanted to design our own binders. So we wanted to design binders for PDL1, which is a very interesting target for immune modulation. And here I show you a schematic of the function of PDL1 in cancer. And basically, when you have T cell encountering a tumor or a cancer cell, it's going to recognize this cancer cell and try to engage with it through the T cell receptor, because the cancer cells usually kind of present aberrant antigens on the surface through the MHC complex. Now, if the tumor cell overexpresses PDL1 and that the T cell is going to encounter PDL1 with PD1, basically it's going to tell the T cell, do not worry, this is a cell from your body, do not engage with that cell. And so the T cell is going to go into what we call T cell exhaustion and apoptosis. So this is why solid tumor microenvironments are very immunosuppressive in most cases. And there is lots of immune checkpoint systems as well. So you imagine that if we make an anti-PDL1 binder, we can block the interaction between PDL1 and PD1 from the T cell and reinstall the T cell activation that would be done with the tumor cell and eventually lead to the apoptosis of the tumor cell. If we look at the structure now of PDL1, so here we're interested in the mouse version because we work with mice models. It's an immunoglobulin-like structure. It's got a conserved exposed hydrophobic patch along these beta strands. And you also got N-glycosylations, which kind of prevent the protein from aggregating in eukaryotic cells. The thing with that is that in E. coli, we don't have glycosylation. So it means that when we overexpress it in E. coli, it forms inclusion bodies from aggregating altogether, which is not so great because the cell-free we are using is derived from E. coli. So the first testing we did, trying to fuse the domain to C1 DNA binding domain, we really quickly saw that this was causing aggregation. So we had activation of the GFP signal in absence of any binder, meaning that PDL1 alone was multimerizing and activating the C1 specific promoter. So we could not use our original system to screen binders for PDL1. We had to adapt and kind of slightly modify the system to make it work. And it's very simple. I'm going to talk you through it. So basically, our idea was to be able to use exogenous PDL1. So PDL1 that is not being produced directly in the cell-free system, but that we can purchase or produce ourselves from mammalian cells. So these PDL1 contains glycosylations. It also contains an Fc domain and is biotinylated. And this is important because biotin specifically interacts with streptavidin. And by kind of complexing these guys together, you can form tetramers of PDL1. And now if you see our cell-free two-hybrid system as some kind of biosensor, you can imagine that these tetramers can be recognized by the binders. And because they're all close together, they're going to drive the multimerization of the binder and the DNA binding domains, and hopefully drive the expression of GFP. So this is what we tested, and you can see on that graph that if we add streptavidin alone into a cell-free system that expresses binders that are specific to mouse PDL1, you see that nothing happens. Now, if we add PDL1 alone, nothing happens. But if we add the complex streptavidin together with PDL1, forming tetramers, now we can see nice levels of activation. So we can sense specifically multimeric PDL1 in our screening system. Now, if we do dose response curve, we can nicely show that we have an increase in response as we increase the doses of PDL1 coupled to streptavidin. And we actually tried this system on different domains. So we tried not only on PDL1, but also on CD47 and CTLA4, and could also witness an increase of the signal as we added more of these proteins complex to streptavidin. So now that we have a fully functional screening system for PDL1, we went on and designed our binders. So we used Bindcraft. So it's one of the papers I showed earlier in this talk. It's a very good pipeline, very easy to use, and very accessible as well. So we sorted the 36 best potential candidates for binding to mouse PDL1, and then we tested them in our screen. So here you can see, similarly to the graph I showed earlier, the kind of response we got adding streptavidin only or streptavidin with PDL1. And you can see that, surprisingly, out of the 36 binders we tested, we had actually quite a good number of hits. And this was really promising. So to go further, we took some candidates and did some SPR to have KD values. So the number 23 had a KD of around 7 nanomolar, and the 35 had a KD of 0.8 nanomolar, which is actually very good. So we know about one thing about designing binders de novo is that they can bind the target, but they can also bind other stuff. Just because we make them sticky, they can bind to other targets maybe. And if you kind of put them in a very complex cellular context, maybe they would not do their job, but they would be distracted by just binding any protein. And so to test this with our kind of most promising target, binder, which is the number 35, we used a cellular assay in which we mimic the kind of system I showed you earlier. In that system, we have T-cell activation is, so this pathway drives the expression of a luciferase gene in Jurkat cells, so it's basically T cells. And on the other side, you have the cancer cell overexpressing mouse PDL1. So the idea is to incubate these two cells together, and if a binder actually binds to PDL1, it should block the interaction between PD1 and PDL1 and restore luminescence, so luciferase expression in the T cell. And this is what you can see on that graph. In blue is our binder. We add increasing amounts of this binder, and you can see that we have increasing amounts of luminescence. And if we do a control with a monoclonal antibody, we obviously have a better response. But the it has 2 binding sites and it's an antibody, so it's very stable. But this was really promising, and it shows that from going from the computer design of binders to the rapid screening in 1 day of protein binders, we can then get really good and functional binders that are suitable for our purpose. So this was actually really cool. Just a side kind of story, because I saw in the program that you guys have an iGEM team as well and are into iGEM. So last year when we published the preprint version of this work, we got contacted by the Heidelberg team that was doing a project on smart synthetic immune cells for precision oncology. And basically they were doing some designs of binder in silico. And you know, iGEM, you have a limited amount of time to do experiments. And so they really wanted to test their designs, but none of the kind of wet lab methods was fast enough for them to screen these guys. So they contacted us, and I shipped them some cell-free and the plasmid and so on. And they did the rest. So I just told them, you know, the basic protocol that I'm using. And within a couple of weeks, they could actually screen their own binders. So here their target was GDF15, and actually had a really good candidate that then tested then in their cellular assay, combining GPCR and binders and so on. And they could see that they could detect specifically GDF15 in their assay with a newly discovered binder. So this was just to show you that we made a method that is actually very simple and very accessible, and students can use, and it's really great to see that it can be used as well than in our lab, and actually be functional as well. So now I'm going to finish with kind of the other avenues we explored for Cell-Free Two-Hybrid. So beyond kind of traditional protein-protein interactions and binder design, we're also interested in small molecules. So we wondered whether we could kind of add into the mix some small molecules to do maybe competitive inhibition, chemically inducible dimerization, and maybe some kind of protein detection, because we saw with the PDL1 streptavidin system that we kind of had something similar to a biosensor in our hands. So to cut a long story short, I'm just going to show you a few examples that we tested. So this takes the BCL2 and LD3 binders that I showed you earlier. And venetoclax is an FDA-approved drug that binds BCL2 with a very good affinity. And our idea was to kind of put these guys together and increasing concentrations of venetoclax and see whether we could displace the complex in our cell-free system. And here you see the results. So in gold is the BCL2 LD3 couple, that with increasing concentrations of venetoclax, we can shut down the activation of the system, so the production of GFP over time. And you can see that with an unrelated system, so the VHH alpha tag, if you add venetoclax, it doesn't do anything to the system. So it's very specific to this protein interaction. So if we go the other way around and try to promote protein-protein interaction, we took the example of a single domain antibody that dimerizes upon addition of caffeine. And we actually took a few mutants as well. So the blue mutant is known to have a better sensitivity for caffeine. And the purple mutant is known to have no more sensitivity for caffeine, so it doesn't dimerize anymore. And you can see that here we not only can sense caffeine that is added into the system, we can also recapitulate the impact of individual mutations on the ability of the system to dimerize. And to finish with the biosensing, we saw with PDL1 and streptavidin that our system was able to detect multimeric proteins. So we wondered with the spike protein from SARS-CoV-2, which is a trimer, whether we could actually detect it if we added it purified into our cell-free system. And basically, if you add a combination of 2 nanobodies that bind on adjacent epitopes of the receptor binding domain of the spike protein, you can get a very quick and nice response only in presence of the spike protein. And this is only happening when you have these two nanobodies together in combination. If you only express one of them, it only forms a trimer of C1, and it seems to not suffice for the generation of signal in the CF2H platform. So we can repurpose our system to screen protein-protein interaction modulation with small molecules. We can also turn this system into a biosensing platform, which is also very exciting. So just to summarize, here we can do many things with that system in the end. We can scan the binding interface, as I showed you, to see which residues are very crucial to the interaction. We can obviously screen protein binders that we designed in silico, and we can do small molecule modulation assays and biosensing applications. So to conclude, I wanted to also expose some limitations of the system. So it's not perfect. We know that our system mostly detects very strong interactions robustly. So interactions that are typically below 100 nanomolar, which for kind of interatomic studies or protein-protein interaction studies for natural protein systems is a bit restrictive. So here we kind of take it as a feature for binder screening because you really want to get all the good binders and you don't really care about the micromolar binders. But it's a limitation of the system. We also saw with PDL1 that expressing proteins that bear post-translational modifications is complicated in cell-free, and they might be difficult to produce as a soluble form. So for that, we kind of implemented a new strategy with the streptavidin and biotinylated PDL1 system. But that's a limitation, so we cannot screen any protein target, and that goes with membrane proteins and so on. And also, what's a bit restrictive is the cost of DNA synthesis, because we order all our fragments from DNA synthesis company. They might have systematic error, which brings kind of the error rate of the system a bit higher. But this is independently of the method you are going to use. You're going to need to buy DNA anyways. So the few perspectives, so continue kind of exploring the cell-free two-hybrid capabilities in cell-free, but also in whole cell systems. Obviously, we have pipelines in which we are developing binders for other applications. And we also want to build upon the knowledge we acquired during this process to build and create new systems with kind of complementary features to the CF2H system. So with this, I would like to thank all of my team members, so Jérôme Bonnet for the guidance, and all the people that actually contributed directly to this work. I would like to thank also the collaborators, Alexis Courbet, Rob Raggett from University of Washington, Jiang, who did the SPR analysis on our binders, and a previous intern that worked really well with me. And also, I would like to thank all the funding agencies in CERN Transfer for their support. And I would like to thank you guys for your attention. I would be super happy to take questions from you.
Wow, thank you so much, Dr. Julien, for the presentation. It's really amazing how powerful and actually revolutionary is this system, the cell-free two-hybrid system. I understand it was created in your laboratory, right, by Jérôme?
Yes, we put it together in our lab for the past couple of years. We've been working on this. Yeah.
Okay, well, thank you so much. I'm gonna pass with the questions now. So first, it was a question of mine. It was regarding about the MD-M2 benchmark.
Yes.
So I don't know if you could show that graphic specifically.
Yes, of course, I can show it. If it lets me. I cannot exit this, apparently.
Oh, no.
So if I do this, well, I cannot go here. I can go one by one. It's fine. Okay.
Yes, yes.
That's the one.
Yes, exactly.
Yes.
Thank you. So I see that there are some proteins that are classified as non-binders by Watson et al. that
Yes.
were classified as binders by your research. So
Yeah.
why is this difference exactly? And is just that these systems, the cell-free system, is more sensitive? Or is this an expected result of the experiment?
So it's a very good question actually, and it's actually not a. It comes essentially from the way we defined binders in both assays. So their method is fundamentally different than compared to our method. So they had to set an arbitrary threshold, saying all the binders or all the candidate that have a BLI signal under a certain threshold, we consider them non-binders, even though they produce a signal. So it's just arbitrary in their terms, but they still class them as non-binders. In our terms, a binder is a protein that produces a signal that is above twofold over the background signal when the binder is added alone in the cell-free system. So if you see some cases where it's a bit ambiguous, for example, this one, we say it's not a binder, but actually it's higher when you have the target, but it didn't reach quite twofold compared to the binder alone. So we say that this one is not a binder, for example. But to kind of come back to the original question, it's just two different definitions of binders. Binders is hard to classify as a kind of binary in binary terms. We just did that for kind of the simplicity of the screening later. But yeah, binder is more like a continuum of affinity. And if you think about useful binders, you kind of want something that's in the nanomolar range, right? Anything above in hundreds of nanomolar, micromolar may not actually be any useful for your purpose. I don't know if that answers.
No, yeah, thank you. That answers it completely. And well, following, we have a question from Marco, just saying that after the sudden change in the lab from wet lab first era to an AI first era, did the skills required to work in the lab change?
So I don't think so, and I think it's quite paradoxical because I think the wet lab kind of screening campaigns and so on are still going on, right? The people that are really good at phage display, they just carry on doing phage display and yeast display and so on. It's just that you need a lot of expertise, a lot of time to these screening campaigns, and they're still very useful. But all the companies you're going to see pop out, all the work you're going to see for kind of precisely designing binders and other type of designs that use AI first principles, you still need at the end of the day experimental validation. So it's not the same type of skills maybe, but now with kind of the speed at which AI can generate designs, experimental validation is still a bottleneck, right? So we can do a lot of metrics in silico and not care about validation in the lab. But I think it's a mistake. So we should do more validation in the lab to understand what is a good binder, what makes a good binder. So trying to understand AI, I think, will not be possible without experimental data. So we still need more experimentalists to do experiments and good quality experiments to be able to understand what the designs we have in our hands are doing.
Thank you so much. That is nice to hear. There was some panic among some areas in science research when all the AI boom got to us. But well, it's nice to hear that the skills are mostly the same around this.
I'm a strong believer that even for AI, right, you need to train AI on data sets, right? So you need actual data in the first place to build models and to make learning protocols. So AlphaFold was built on the PDB. You can have, some companies are building their own data sets to curate and have very good, really good resolution data, to only have a very curated databases. But yeah, I think you still need a lot of experimental validation and experimental work to go hand in hand with AI to kind of boost up what we can do in the lab and outside the lab.
Well, thank you. Just continuing, Alejandra is asking that, do you see CF2H being used in early stage cancer therapeutic discovery?
That's a good question. I don't know. We kind of made this system to be very simple, so does not give as much resolution as BLI or SPR, for example. But it's also very cheap. So if you want to kind of do a first screening round, I think going for a cell-free system to express proteins and test their affinity, and CF2H actually has a lot of potential for that. So I think it could be a good, as we show with PTL1 example, right? So this is directly linked to cancer therapeutics. But I think it has also potential for small molecules and other things that we are working on right now.
All right, thank you. The next question is by Alexander Wilber. It's he asking, how can CF2H account for variability in signal intensity caused by difference in protein expression, like levels, solubility, and stability?
Yes, so that's a very good question. We actually addressed that a bit deeper in the kind of revised version of the paper. So this work now has been accepted for publication. It should be coming out in a few days. And yeah, some of the concerns were about how can you kind of embed, protein expression signals, like variations in protein expression and solubility into the signal you get. What we did was experiments doing Western blotting on all the binders that you see here. For example, all of the NDN2 binders, we did blots on them to see their expression levels. And what we found was that we could not correlate high signal with high protein production. And we could not correlate lack of signal with weak protein expression or solubility. What we could see is that there was examples of everything. So if a protein is very well expressed and has a really good affinity, it's going to produce a very good signal. But if a protein is not expressed that well and still has a very strong affinity, it's still going to produce a very strong signal. And we think that the difference in expression and solubility between these binders is not that huge when you think about it in terms of you think there is probably 2 to 5-fold differences in expression. So that's still a linear scale, right, in terms of difference. But if you think in terms of affinity, you're more thinking in log scale, right? So if you have a picomolar binder, having a 2 to 5-fold lower expression or solubility than a different binder is still going to produce much more signal than another one with micromolar affinity. And so this is our conclusion on that. So we were first kind of worried about this, but in the end, we kind of see it as a feature as well. So our signal provides a kind of comprehensive, kind of captures all the features of the binder. So if it's well expressed, if it's binding well, and if it's soluble, it gives a good signal, and that's probably your best bet for your next candidate. So that's why we see it kind of as a feature of our screening.
All right. Then the next question is by Micaela de Fatima. She's asking why the E. coli bacteria was used and why not use other kind.
That's a good question. So typically, cell-free systems, so TXTL systems, have been mostly developed with E. coli. I know a lot of groups working on cell-free are trying to make cell-free from other organisms because they have different properties. There is cell-free from eukaryotic systems, so there is insect cell, plant, human cells, CHO cells, and so on. It's just that they do not have the same properties, they do not have the same yields, and the same cost as well. So we know that TXTL is the cheapest because it's just bacterial lysate. You can have more expensive cell-free systems, eukaryotic ones that are much harder to produce, and you'll get not very good yields in general. So for us, it was just because we had the kind of expertise in the lab of building cell-free, making cell-free TXTL from E. coli, and also the strain that we use is compatible with linear DNA. So other system might not be. Yeah, that kind of answers the question, I hope.
It's also, maybe it also helps that one of the properties for this cell-free system is that it's very quick. So maybe the E. coli helps for that actually as well, right?
Yeah, so in prokaryotic cells, in E. coli, the transcription and translation are kind of linked together. So you have transcription going on, and then ribosomes straight away start translating your protein. So that's why we see responses in a matter of half an hour with our system, which is amazing. And basically, we let the reaction run for 12 hours just for the sake of having the plateau at the end. But just passing by the plate reader and looking at your plate after an hour and a half, you already have an idea of the good binders, essentially. So yeah, I think that's a good point. Yeah, it's really fast.
All right. And then Sasha Constante has another question, which is, your system seems to detect protein-protein interactions very quickly. What are the main limitations in terms of sensitivity or false positives compared with traditional methods like yeast 2-hybrid or biophysical techniques such as biolayer interferometry?
Yeah, so that's a good question. We are kind of limited with our system to kind of high affinity, good affinities, so low KD. So what I said just at the end is that anything above 100 nanomolar, we cannot say we can reliably detect it. We might be able to detect it just fine, depending on the interaction, the nature of the protein itself. But we might miss it. And this is kind of problematic for doing basic research, let's say on protein-protein interactions, because if you're not reliably catching events of interaction that are above 100 nanomolar, then it's most of the proteins that actually interact together in nature that you are going to miss out. So for binder screening, I think it's a good thing because we can select the top binders right away. But for kind of interactomic studies, I think 2-hybrid methods like yeast 2-hybrid or BLI, if you can afford. BLI is cheaper than SPR, is faster as well. So you can, it's better to do BLI for that. But then it requires you to have your purified proteins. If you have mutants, you need to purify all these mutants and so on. And yeah, there was another part about false positives. No, I think,
yes.
Yeah. So false positive, we didn't see much false positives in our system just because of the fact that it only works with very high affinity proteins. So if you have kind of passive weak interactions or non-specific interactions, you don't tend to catch them in the system. So that's actually a great feature for binder screening. So you don't get distracted by false positives. At least we didn't see any. The only cases where we saw a signal activation that was problematic with the screening is when we tried to fuse PDL1 directly to C1, and we saw aggregation of PDL1, which was causing the promoter to actually activate itself. So that was the only case where we had issues.
Okay, thank you again. Following, Ivanova Castello has asked, since you mentioned that the reactions can be performed in very small volumes and high throughput formats like the 384 well plates, how scalable is the system for screening thousands of variants in a directed evolution workflow?
I would say it's just scalable. It just depends how many candidates you want to screen. I think the main limit is going to be your budget, because here we're not working in kind of a single tube with a library, which is usually more affordable than ordering genes separately into different wells. So in terms of scalability, if you have multiple plate readers and so on, you just can scale up to thousands of reactions per day. You could also couple it to robots and do liquid handling robots and do kind of a fully automated workflow. So then you wouldn't even have to do anything in the lab. You can just program the robots to do all the pipetting, and you would just launch the plate readers. So I think it's really scalable. It's really tiny reactions. So it's really convenient. But yeah, the main restriction I would say for such studies is the budget. So ordering genes right now it costs around 7 cents per base pair. And so if you have, 384 genes to order that are 1 kb, it's going to be a bit of a budget.
And that cost is also, like here in Peru, is approximately the same. So I think it's
Okay.
a global price, right?
Yeah. So unless you are a really big lab, which passes through a lot of these plates during the year and you can get a deal, it's around 7 cents. So hopefully it would go down in the coming years so we can all order more DNA sequences and test stuff more rapidly.
Yes. Hopefully. And then continuing, Maricielo Maita says, first of all, congratulations for the work. It was really interesting, especially how fast the system is. And I was wondering if a cell-free system could eventually replace some traditional protein-protein interaction assays in early stages, or if you see its main role more as a pre-screening test for large libraries of designed binders. Thanks.
So I think both are valid. You can definitely use cell-free systems, and in the coming years, I expect a lot more people using cell-free actually in the labs, and we see a lot more companies actually harnessing cell-free to kind of prototype constructs before using them in E. coli, for example. So expression constructs. If you need to use different tags, different versions of promoters, and so on, it can be quite painful to test all of this in live cells, so in whole cells. So using cell-free for prototyping purposes, for screening purposes, for binders especially, I think is a very good idea because it's very small volumes, costs basically nothing, and is very scalable, as we mentioned before. Now, is it going to replace actual methods? Well, you cannot get association, dissociation constants, and so on with basic cell-free methods, at least with our screen. So we still rely on SPR, BLI, and so on to get real affinity values. And I think we still need this very much. But I think totally for a pre-screen, and I think I see it also as when you have very large screening campaigns, so people screening billions of variants, sometimes they have like 100 or 200 hits with their display methods. So how do you select the good ones? Maybe you just need one. So how do you go on and test all these 200 variants? Then you can use cell-free. after your first initial massive screen, you can use Cell-Free to distinguish, subtle differences between your binders, and maybe select the top ones that you can then characterize, do SPR and cellular assays on them. So I think that's answering the question pretty much.
Yeah, it does. All right, so just following, then we see the question from Angie Avalos. Do you see this system being integrated with computational protein design pipelines to rapidly screen large libraries of binders? For example, those generated by AI-based protein design tools?
I mean, that's kind of what we do here in this work. I guess, and what would be nice, and what I think you kind of touch upon with this question, is having a fully embedded system in which we train a model that generate binders, and we can directly feed the results of our screening campaign back into the model to try to teach it what worked and what didn't work. And if we can do that, we can get smarter and smarter at designing proteins using these models. And I think that would be very great, actually, to kind of build systems and smarter AI systems that would be trained for a specific task. I think that's a good idea, yeah.
All right, thank you again. And now we have Max Salvatierra asking, what type of reporting genes do you typically use, and which one provide the greatest sensitivity? And also, in the case of protein RNA binding, how can we assess whether or not they interact using analogous synthetic biology systems specifically?
Interesting question. So for the RNA part, I guess if your protein binds to a specific RNA sequence or an RNA structure, you would need to, first of all, depends if your protein is a monomer or a multimer and so on. But essentially, if you fuse your protein to C1, DNA binding domain, and have multiple time the sequence that this protein is bound to in your RNA sequence, then you can multimerize it directly on the same RNA molecule and try to sense it using the Cell-Free system. So I don't know if that answers that part of the question. The first part was, I can't remember, sorry.
Oh, about the reporting genes, which
one? Oh yeah, the reporter genes. So we tried here SFGFP, which we use, and which is quite good because we can follow it live. So as I was saying, you just go in front of the plate reader and see already the wells that starts producing GFP after a couple of, 30, 35 minutes. We tried nanolook, so luciferase reporter genes. These are also quite good, but in terms of signal to noise ratio, it was very similar to what we had with our SFGFP system. So we had much more signal, but we also had much more background. So it probably means that we would have needed to re-optimize all the concentrations of plasmids that we use in our system, the concentration of linear DNA for the binders and so on, to make it compatible with luciferase and try to get a better signal to noise ratio. The thing with luciferase is that it requires more kind of hands-on time, because you need to add the substrate at a given time and then do the readout. And so this means that you cannot follow live the production of luminescence over time with the nanolook. You need to add the substrate, which kills the Cell-Free reaction to reveal it. So it's just an endpoint measurement. So you can miss on certain aspects of the kinetics of production.
But you could use a luciferase as at the end, right?
Yeah, exactly. You can use luciferase. We did it, but we did a time point right at the end of the cycle of CF2H.
All right, thank you again. Now another question from Alonso Quinto. He says, hi, what were the main difficulties of the project, when you were working these de novo design proteins specifically?
So the main difficulties that we encountered, some unexpected ones. So I think it's good that I tell you that, guys, but what I wasn't aware when I started this was that I started doing everything with linear DNA fragments. So we were just buying plates. And for the first proof of principle examples that I showed with M-cherry and so on, I ordered them as DNA fragments. And here you can see on this MDM2 binding set, actually, that some are highlighted in pink. So these actually, if you look at them in this state, they are not classified as binders. But we found it funny that we had so many differences with the results from Watson et al. So we went and tried to clone these sequences. And while I tried to clone them, I could never get the right sequence. There was always mutations located in the same area of the gene that were preventing the right sequence to assemble into that plasmid. And it was not toxicity because then I kind of repaired these mutations using primers, and I could have the fully assembled plasmid right away. And when we tested them again in a second round, amplified, still using PCR from sequenced plasmids, then we could classify them as binders. So it means that in the plates we receive from DNA synthesis companies, there was systematic mistakes in some wells that we received. So this is part also of the limitations of this, is that we do not have any sequencing, culturing, cloning steps, but we rely on the quality of the DNA that we receive. And so if you have 100% of your sequence that is mutated in your well, and it still passes the QC, then it's not going to be a hit, but it might not be because it's actually not a binder, but it might be because it's actually not expressed. And this is why we are doing a combination with blotting. So we add a little tag at the end of the binders to be able to do blots to check their expression. And the other question, so sorry, there was one part of the question and the other one was?
Oh yeah, I think the question also asks like any difficulties working with de novo proteins?
De novo proteins?
Yes.
Not really. Actually, we were quite surprised, at least with the ones that we designed for PDL1, had a really good success rate. Then when we went on and put them in E. coli to express them, they were also really easy to purify, highly soluble. And for all these de novo design proteins, they generally go through MPNN, protein MPNN pipelines, which, it's the inverse folding step, which tend to yield very highly soluble and expressible proteins. So they're very stable and very easy to work with. Then the pipelines themselves, for designing proteins, we just installed Bindcraft from the GitHub repo of Martin Paquesa and just started designing. It was very easy.
And just one small question from me again. Among all these programs, like related to Minecraft, did you try any other alternatives, and which one was better?
So right now we are kind of exploring other type of pipelines. So we are using RF diffusion as well for some projects. We are using Bindcraft for some others. And we are also kind of playing with different type of settings for RF diffusion. And we're also trying to do some designs with just protein and PNN and see what happens. So we're in the process of testing all this in the lab. So I cannot answer your question on which one is better than the other, but I think they all have their strength. So I think Bindcraft is amazing because you can get really good affinity proteins right away in one shot. But for some specific targets, it might not yield any protein at all. You might even struggle to propose viable candidates. So then you have to either change the parameters or turn to a different kind of RF diffusion style method because it's struggling too much. this is all I can say for now on what we have been testing. But we should have more going on because we are installing a bigger server. So we should be able to run all this smoothly in our lab.
Yeah, and I think the testing will take some time because there are also new models like Bolt's gene,
Yes.
and they have some mixed results, but yeah, there are many to try.
Exactly, yeah. I don't know if you saw the adaptive competition results, and actually a lot of Bolt's-Gen designs cannot pass the filters, but we're not binding at all experimentally. So I think that's really interesting as well. So that tells you the difference between making a pipeline that can generate designs with very good metrics and yet failing experimentally. So I think these competitions are really cool and really good because it shows how much we require experimental validation. And we need new method for screening these binders, easier methods for people that are maybe not specialists to be able to play around on the computer, but also test these guys in the lab for their research.
Yeah, completely. For example, here there was a competition for making a binder for the Nipah virus. I don't know if you've heard of it.
Yes.
Yeah, and specifically the ones that were produced with Bolt's gene, we expected to get like great results because it's one of the latest, but it had one of the worst results specifically. So, and Minecraft had well, very good results as well. So I think it's like it depends on the project as well to use specific models, and still we should use many models and compare them each other for every project in itself.
Yeah, and that's why I think it's good to compare them and to have kind of rapid methods to be able to screen in the lab. So you say, oh, I'm going to do 24 designs with, I'm going to screen 24 designs I did with Minecraft, 24 with Bolt's-Gen, or 96 for each or whatever. So you need something to be able to screen them rapidly and also to be able to iterate quickly. So if you have your results within one day and you have kind of some settings in the pipeline that you tuned and you know these binders that worked were done with these settings, then you can take these settings and do another round using these settings and try to hope to get better binders or with different specificity and so on. So I think it's why we need kind of easy processes to go fast. They need to be robust, obviously, but so we can have the cycle of design, build, test, learn going on.
Well, thank you so much, Dr. Julien. We have 10 minutes, so I'm gonna ask one final question, which is from Roberto Cabezas. He asked, which biomedical or pharmaceutical applications do you think could benefit the most from platforms like CF2H?
Well, that's a good question. I think any application that requires binder discovery, I think it would be beneficial because it's fast, it's affordable, cell-free costs nothing, as I said before. So cancer research is something we kind of working in. Working in the team using SynBio tools, and this is one of these tools that kind of can be multi-purpose. So I think there is not an application in particular. Now that we have the tools, we open the door to kind of new possibilities. So maybe there is ideas that some people would have with this system that I didn't even think of yet. And this is the point of it as well, is to just being able to ship that method to as many labs as possible. People teach people how to do cell-free and try to make it accessible. So the guys doing the, building the computational pipelines, I think did a great job of making it open access, making it accessible through collabs and so on. And so we need to do the same with experimental methods. So if you have a method that you're the only one to master, yes, it gives you kind of a monopoly on what you can do with it, but being able to make a very simple, robust method that you can kind of distribute around and be used around, I think it's much more valuable.
Oh, well, that was the last question. Thank you so much, Dr. Julien. It was a great honor and a great pleasure to have you here with us in the symposium. And well, I just gotta say it's an amazing, really research, and amazing work you're doing and with your group. And well, that will be all actually. Right now we have another presentation in approximately 30 minutes with the Dr. Anna Zuniga. So, well, thank you. So first of all, thank you all of you for your questions and for making the session a great start. We want to express our sincere gratitude to the Dr. Julien for his presentation and his kind answers. And well, yeah, we will invite you all to wait just 30 minutes and we will come back now with the presentation from Dr. Zuniga. So thank you again. And
Thank you. Thank you,
yes,
guys.
it was amazing, really. Thank you.
Bye.